Hi everyone, I'm Natalie, and today I would like to give you an introduction of skinomics, the bioinformatics in skin biology and dermatology. So today you will be able to understand the definition and development of skinomics, an overview of DNA microarrays and their role in skinomics, as well as a case study of melanoma and DNA microarrays. So let's start with an introduction of skinomics. But before diving deeper into skinomics, I'll need to first start with introducing the omics techniques. Omics techniques have revolutionized the way we study biological systems, providing a holistic view of complex processes. Rather than focusing on a single protein or a gene, omics techniques comprehensively deal with the entire collection of all proteins or the whole genome. These methods generate massive data sets that require bioinformatics to process and draw meaningful conclusions. Thus, bioinformatics plays a critical role in processing and interpreting these massive data sets, acting as the backbone of omics techniques. And skin was among the first targets analyzed using omics. And dermatology embraced the approaches very early as it is very easily accessible. Then we have skinomics, which is a specialized application of omics techniques. So, as you can see here, apparently skinomics is skin biology plus bioinformatics. It refers to the bioinformatics studies specifically focused on dermatology and skin biology. It aims to expand our understanding of skin biology, improve the function of healthy skin, and assist in treating skin related diseases. Moving on to our next part, I would like to introduce you the DNA microarrays analysis. It is the most frequent and continual methodology in skinomics. So the first step for microarray analysis is the sample preparation. In this example, we have samples 1 and 2. Sample 1 is a tissue from a healthy cell, and sample 2 is a cell from tumor tissue. First, the RNA is extracted from both samples and transcribed into cDNA using reverse transcription. During this step, the cDNA is fluorescently labeled. So cDNA from the health sample 1 is in green, and cDNA from the malignant sample 2 is in red. After sample preparation, let's take a closer look at the microarray plate or chip. So, a microarray plate has many spots. And looking more closer into one of them, we can observe that each spot is coated with multiple identical probes. These are short oligonucleotides that cover the sequence of the specific gene. Now we can observe the spot of these of the gene X when we add our samples. In this example, molecules from sample 1 hybridize with the probe from that spot. But sample 2 does not contain complementary cDNA molecules and is washed away. So the spot we were looking at appears green after laser scanning, which tells us that, oh, gene X is expressed in sample 1 but not in sample 2. Next, let's examine a different spot on the microarray containing different oligonucleotides against gene Y. We might observe the opposite scenario with only sample 2 containing the complementary cDNA but not sample 1. In this case, the spot of gene Y appears red after scanning because of this specific fluorescent label. As a conclusion, gene Y is not expressed in sample 1 but in sample 2. The third possible scenario is that both samples contain the molecules that may hybridize with a gene's probe, as shown here for a hypothetical gene Z. After laser scanning, the spot of gene Z looks yellow because both fluorescently labeled samples are present after washing. These results tell us that gene Z is expressed in both sample 1 and sample 2. In reality, the cDNA sample is added to the whole plate in one step. During microarray chip analysis, the laser excites the fluorescent dye and the emission levels are measured by a detector. Raw data can then be analyzed using bioinformatics, and this is the overall workflow for DNA microarray analysis. Moving on to the next part, the most advanced and most important microarray studies in dermatology concern melanomas. So here I want to talk about a case study of melanoma and DNA microarrays. Melanoma is among the most aggressive human cancers and was one of the earliest targets of DNA microarray studies. The DNA microarrays have been used in, firstly, comparison of metastatic and non-metastatic melanomas. Secondly, in the integration of transcriptional and genetic data in melanoma. And lastly, in the development of non-invasive sample collection from melanomas and pigmented lesions. The modular progression of melanoma transitions from benign pigmented nevi to primary melanoma and eventually to metastatic melanoma. Researchers use transcriptome analysis and laser capture microscopy to study gene expression in different stages of melanoma. The studies identified genes that can accurately distinguish between normal skin, nevi, primary melanomas, and also two types of metastatic melanomas. Interestingly, metastatic melanomas exhibit two distinct gene expression patterns, resembling either the flat or the nodular components of large primary melanomas. Additionally, hundreds of unique molecular markers specific to the malignancy were discovered, providing insights into melanoma progression and also the potential diagnostic targets. So, this is all for my presentation today, and I hope you have a deeper understanding of what is skinomics. And thank you for listening.